Thank you for joining today's Akoya Academy presentation titled Custom Conjugation and Antibody Panel Design. I will begin with a brief overview of the CODEX technology and do a deep dive into custom conjugation. Then I'll review CODEX antibody panel design tools and wrap up with an example of a customer panel. Let's start with a CODEX technology overview. According to my customers, CODEX is the ultimate discovery tool. In 2019, the scientists named CODEX one of the top 10 innovations of the year. With a CODEX, you can look at over 40 biomarkers within a single tissue sample. It's compatible with both FFPE and fresh frozen tissues, and you can even image TMAs. Our proprietary chemistry is gentle and does not ablate or destroy the tissue. And our flexible custom conjugation option allows for researchers to use any combination of antibodies either from Akoya or outside Akoya to explore your tissue of interest. In summary, the high level of multiplexing with the CODEX solution allows for a deeper interrogation of your tissue microenvironment with the potential to accelerate biomarker discovery. The CODEX system is a part of a complete end-to-end solution that includes optimized antibodies, reagents, fluidics control, and a software suite for downstream image processing and analysis. So how does the CODEX accomplish such ultra-high multiplexing? Well, the key is in our proprietary chemistry. CODEX stands for co-detection by indexing. Each antibody in a CODEX panel is conjugated to a unique oligonucleotide barcode. So if you want to look at, say, 40 different antibodies, you would have 40 unique barcodes attached to them, and all 30 antibodies will be applied to the tissue all at once. Then we have dye-labeled reporters, which have the complementary oligonucleotide sequence with a fluorescent dye. This barcoding system is what enables the highly specific detection of markers within your tissue. The reason we use just 3 fluorophores is because they're spectrally distinct. What this means is spectral unmixing is not required during downstream image analysis. Now, when the barcoded antibody and the reporter with the complementary oligonucleotide are incubated together, they hybridize. The resulting fluorescent signal can be captured for each biomarker. Now, once these 3 markers are imaged, just the reporter is washed away, and then 3 more reporters are applied to the tissue to reveal 3 new markers. We'll continue this process in a cyclical fashion until we reveal your entire panel of markers. So how do we accomplish this cyclic revealing of antibodies? Well, we do so by using the CODEX instrument that integrates with an inverted microscope. And the CODEX will facilitate the reveal image remove process during your multi-cycle experiment. Now, each barcode has a specific dye-conjugated reporter associated with it, and the antibody barcode distribution can be seen on the left. This chart tells you which barcodes have inventoried CODEX antibodies associated with it. Now on the right, you'll find the CODEX reporter distribution, and you can find what reporters are associated with a barcode by matching the numerical pairs. So for example, the corresponding reporter for barcode BX001 is RX001. And the reporter RX001 has an AF488 or an AF750 version. There are 2 groups of fluorophores we suggest using depending on the sample type you would like to study. And both groupings, 1 and 2, include DAPI. For fresh frozen tissues, we recommend using AF488, ADO550, and Cy5. For FFPE tissues, we recommend using AF750 instead of AF488 to counter high levels of autofluorescence generally found in these tissues. Now that we're all caught up on the CODEX chemistry, let's talk about antibodies. So how does one decide what antibodies to use? Well, we first recommend one visit the website and see what pre-conjugated antibodies are available for sale through Akoya. As you can see here, we have a wide offering of human FFPE antibodies and human and mouse fresh frozen antibodies. CODEX inventoried antibodies are extensively validated by Akoya R&D and sold by Akoya. You can find the list of inventoried antibodies on akoyabio.com. Now, what can you do if your antibodies of interest are not sold by Akoya? We have a solution for that. What you can do is custom conjugate your own markers of interest. This next section will provide guidance on just that. So the first step in custom conjugating your own antibodies is purchasing a purified antibody from a non-Akoya vendor. Then you'll purchase an activated barcode and a conjugation kit through Akoya and perform a conjugation reaction to generate a custom conjugated antibody. These antibodies can now be used in a CODEX experiment. Please note that the purchase of an activated barcode includes 2 vials of reporter. Choosing a good antibody clone is the key to a successful custom conjugation. We have a couple of resources available for you to provide some guidance on selecting a clone. First, we have the CODEX screened antibody list. These are antibodies that we were able to successfully conjugate in Akoya R&D, but we do not sell them. Instead, we provide information regarding the target antigen, the antibody clone, which vendor the antibody was purchased from, which tissue was screened, and what antigen retrieval conditions were used if applicable. You can also refer to our community antibody list, which includes antibodies that were successfully conjugated and tested and shared by CODEX users. If you can't find your clone on either one of these lists, then you can refer to the guidance on the next 2 slides. When purchasing a purified antibody, you can purchase them through your vendor of choice, but please note we strongly recommend that they are in PBS or a similar buffer without the presence of carrier proteins like BSA. If you can't find an antibody preparation in pure PBS, then we recommend using a commercially available kit to remove the BSA and purify your antibody. Sodium azide does not need to be removed and will not interfere with the conjugation, and use of lyophilized antibodies are strongly discouraged. Use these only after exhausting all other options. Now, it's very important to note that you must always measure the concentration of your commercially purchased antibodies using a nanodrop. And the reason for this is the concentrations that are listed on the tubes are usually not accurate, and using the wrong amount of antibody could result in a poor conjugation efficiency. Now, when you're choosing a clone, please choose one that has been demonstrated to work on your tissue of interest. And a great place to start is to do a literature search. In our hands, we found that IgG clones have been more successful than IgM. And you want to validate the clone prior to performing a conjugation reaction. What this means is you must identify a positive control tissue, perform a standard immunofluorescence or IHC experiment to identify positive staining, and then you want to optimize the staining conditions and antigen retrieval conditions if applicable. And finally, you want to validate the staining specificity prior to proceeding with a conjugation reaction. Generally speaking, if your antigen is less abundant in your tissue, it will produce a low intensity signal. Thus, for low abundance antigens in fresh frozen tissue, we recommend reporters with either Cy5 or ATO550. For FFPE tissues, we recommend Cy5. Now, on the other hand, if your antigen is more abundant in your tissue, it will produce a high intensity signal. So for fresh frozen tissue, we recommend using Electrophoresis 488, and for FFPE, we recommend using ATO550 or AF750. Now, if you're using AF750, we suggest performing a preliminary screening on a different channel. And this extra step is recommended as camera sensitivities tend to decrease approaching the near-infrared region. And comparing the staining given by the same marker on a different channel might be helpful for establishing optimal exposure times. Here's the pre-experiment checklist, and this details all of the material you'll need to perform the custom conjugation reaction. To start with, you'll need 50 micrograms of purified antibody. You'll also need 50 kilodalton molecular weight cutoff filters. Please make sure that you use the recommended size cutoff. If you use a higher or smaller filter size, your conjugation will not be successful. Please be sure to also use nuclease-free molecular biology grade water because we are using DNA oligos. As far as instrumentation, you will need a centrifuge, a nanodrop, and a vortex. When you purchase a conjugation kit, you'll receive 2 shipments, one that goes at 4 degrees and one that goes at -20. Please note the conjugation does not include the barcode or the reporters. Those need to be determined by you and purchased separately. In this slide, we'll discuss the CODEX conjugation process step by step. The entire process takes about 4.5 hours from start to finish. There are 2 incubation steps, one 30 minutes and one of 2 hours. And please note that the recommended quantity of antibody, again, is 50 micrograms. Please do not change this amount, as this is the optimized amount needed for the reaction. So the first step is to assign a CODEX barcode to each antibody. And then you'll block the filter. After that, you'll measure the protein concentration with a nanodrop and calculate the volume of solution corresponding to 50 micrograms of antibody. Next, you'll purify the antibody solution and perform a reduction reaction. This reduction reaction reveals the thiol groups to which the barcodes will then attach. Please note that this incubation time is very important and it's critical not to exceed 30 minutes. Exceeding 30 minutes will result in irreversible damage to antibodies and an unsuccessful conjugation. Once this incubation is complete, you'll proceed to conduct a buffer exchange, and you can prepare the barcode solution at the same time. Now, once that's complete, you'll set up the antibody conjugation reaction, which will take 2 hours. Please be sure at the end of the 2 hours to set aside 5 microliters of the solution for quality control, and you can keep this aliquot at 4 degrees centigrade. Proceed with the purification step, which consists of 3 washes and spins. Please do not abbreviate this process. And finally, after these spins are complete, you'll collect the CODEX antibody solution and add storage solution. At this point, your antibody can be stored at 4 degrees between 6 months to a year, and the conjugation process is complete. The antibodies are ready to use. Please wait 2 days before validating the newly conjugated antibody. Now, once you've performed the conjugation reaction, the next step is to perform quality control and antibody validation. So this is a 2-part process. The first part is to perform a protein gel electrophoresis, and the second is to perform the antibody validation, which consists of comparing custom conjugated antibodies with direct dye antibodies, identifying the correct titration at recommended exposure times, and determining the specificity of the staining. These steps are also done by Akoya R&D to validate all of our inventoried antibodies. Here's an example of the first step. So, if the conjugation reaction is successful, you will see an increase in the heavy chain of the antibody. In this example, you can see the protein ladder on the left and the unconjugated control antibody on the right. Next to the unconjugated antibody, you can see a successfully conjugated antibody. The molecular weight of the heavy chain is higher than that of the unconjugated control. Please note that this procedure only indicates the success of the chemical reaction. It is vital to perform the antibody validation, which is highlighted in the next steps, to verify correct staining in tissue samples. The second step of QC is the validation of the conjugated antibodies. We want to ensure that the CODEX antibodies demonstrate equivalent staining patterns when compared to dye-conjugated antibodies. This slide shows the tissue architecture and the follicle region that you would expect to see stained with B220 and TCR beta. B220 is in red and TCR beta is in green. When CODEX barcoded antibodies are compared to their dye-conjugated counterparts, the correct staining pattern is observed and the overall cell distribution is consistent. Regions of interest are shown in white, dotted squares, and zooming into those selected regions show that the cell morphology is visible and it displays the expected staining pattern. The next step is to optimize the titration of antibody you use to stain your tissue. On this chart, you can see which titrations are suggested for fresh frozen tissue and FFPE tissue. When you're imaging tissue stained with these titrations, please start by using the recommended exposure time seen here for the given channel. You can optimize these if needed, but for background subtraction purposes, it is recommended to use a similar exposure time for a given channel across your entire antibody panel. Here you can see a range of titrations tested for the custom conjugated antibody. In this case, it's CD45 in fresh frozen mouse spleen. An optimal titer is selected based on maximal signal-to-noise ratio and average signal strength. We also assess the images to make sure there is minimal non-specific staining and signal saturation. Regions of interest are selected based on tissue quality, making sure to avoid areas of tissue with rips or autofluorescence. In this specific example, the signal is strongest at a titer of 1 to 250, and the best signal-to-noise ratio is also observed at that titer. Here's an example of MHC2. In this case, 1 to 250 is saturated, and the signal at 1 to 1000 is too low. The 1 to 750 titer gives a good signal with much improved signal-to-noise ratio. So the take-home message is, when assessing titrations, it is very important to take into account both the signal and the signal-to-noise ratio to determine which titration to use for a given antibody. Now, once you've achieved an optimal titration, we recommend using a positive and negative counterstain to validate the specificity of your newly conjugated antibody. In this case, our newly conjugated antibody is CD4, and it's shown here in green. A positive counterstain is an antibody you can expect your newly conjugated antibody to co-localize with. In this case, CD4 should co-localize with TCR beta, which is seen in blue. A negative counterstain is an antibody for which you would not expect your newly conjugated antibody to have any overlap with. In this case, CD4 should not overlap with B220 visualized in red. And that's exactly what we can see here in this image. CD4 staining can be confirmed by co-localization with TCR beta and no overlap with B220. Internally, we also test our newly conjugated antibodies in a CODEX multicycle experiment. You can see here that the CD4 staining pattern, as seen in the previous slide, is conserved in the multicycle experiment. This next section of the talk will cover CODEX panel design. We have a number of tools available to aid in designing your own antibody panels. This antibody panel design tool is available upon request, and it can help you answer such questions as which barcode and reporter should I use for my custom conjugation, and which antibodies can I include in my panel. In the next few slides, I'm going to break down each tab of this design tool. The first tab gives you a summary of the barcodes available and whether these barcodes are used in our inventoried panels. This information can be garnered from the chart on the left. The chart to the right outlines a corresponding reporter and which fluorophores are associated with that reporter. Let's highlight some examples. First, all of our barcodes are available for custom conjugation. I can ask, for example, if there are any human FFPE antibodies that are already conjugated to BX007 in Akoya's inventoried list. And what I do is I simply look for the column that says human FFPE, and I scroll down to BX007. I can see here that there's already an inventoried antibody associated with BX007, and moreover, if I look to the next chart over, I can see that the complementary reporter is RX007, and it is available in two configurations, AF488 and AF750. For our next example, I want to learn which Cy5 associated barcode does not have an inventoried antibody assigned to it for human FFPE. So first, I try to find which barcodes are associated with Cy5, and I look under human FFPE. Next, I scroll through the list and I see that BX027 is open and associated with Cy5. In other words, there is currently no inventoried human FFPE antibody associated with this barcode, and I can use it for custom conjugation if I like. The second tab in our antibody design tool has a list of all of the CODEX inventoried antibodies. Please note that all of the column headers are sortable, which makes it very easy to find the information you are looking for. Let's look at a couple of examples. So I noticed on the summary tab that there is a human fresh frozen antibody inventoried for BX003. Which antibody is that? Well, first I can sort by sample type to only bring up my human fresh frozen antibodies. Then I can look for barcode BX003, and looking across the row, I can see that CD19 is conjugated to BX003. The part number can also be found here in case you would like to purchase this antibody. Another question I could ask is, say I have space on my mouse fresh frozen panel for another ADO550 antibody. What inventoried antibodies are available for ADO550? So how would I approach this? Well, first I would sort by sample type, and then I would sort by reporter dye. This would display the 7 ADO550 associated antibodies in our inventoried antibody list. These include CD2, keratin 14, CD3, and others. The final 3 tabs in our antibody design tool is a place for you to organize your antibody panel, which can include both inventoried and custom conjugated antibodies. So if you were to ask, I have a list of markers for human fresh frozen samples, how do I go about designing my panel? You would use this particular tab. You'll see there's a tab for human FFPE, human fresh frozen, and mouse fresh frozen. So in the case of this example, you would choose the human fresh frozen tab. First, you would add the inventoried antibodies that you want to include in your panel. And in the next column, you would include the custom conjugated clones that you are including in your panel. Be sure to include them under the correct barcode. In this example, we see that CD63 is a custom conjugated antibody assigned to BX013, which is associated to AF488 reporter RX013. So this is how you would, read this particular tool. On the bottom of this list, you can see the totals. There are 23 markers in this panel, and there are 9 ATO550 reporters, 6 Sci5 reporters, and 8 AF488 reporters. If possible, you want to try and keep the dyes evenly distributed. In this case, if I were to seek to add any more custom conjugated antibodies to my panel, I would try to conjugate them to a barcode associated with AF488 or a Sci5 reporter. If I were to add more ATO550 associated conjugates, then my experimental runtime would increase. I'll highlight this again in this next slide. Now that we have used the panel design tool to organize our panel, we can see how our panel translates to a CODEX multicycle experiment. Of course, the first and last cycles are blank cycles, and these are used for background subtraction purposes. Remember, CODEX reveals 3 markers at a time plus DAPI in a cyclic fashion. Therefore, each cycle includes 4 distinct spectrally unique fluorophores. So cycle 2 will reveal CD45, CD2, and CD3, and cycle 3 will reveal CD8, CD104, and CD4, and so on. In cycle 10, since we have fewer 488 and Sci5 reporters in our panel, these positions are left empty. Balancing your panel across the different dyes means that you can run fewer cycles and have shorter overall run times. During the last section of my talk, I will describe a panel designed by one of our wonderful CODEX users, Dr. Diane Saunders of Vanderbilt University. Her experimental goals are to examine cell niches during development and diabetes pathophysiology, and she is using the CODEX technology to explore cell-to-cell interactions in the developing human pancreas. In her hands, she found the conjugation protocol quite straightforward and recommends choosing barcodes for custom conjugation after determining which pre-conjugated inventoried CODEX antibodies will be included in your panel. She reports having successfully conjugated 8 out of 11 antibodies, and all of these antibodies were monoclonal and free of carrier proteins. Here is Diane's CODEX panel for understanding the pancreatic tissue microenvironment. As you can see here, she has a mix of inventoried antibodies highlighted in blue and custom conjugated antibodies visualized in pink. About half her panel was custom conjugated. Next, I will perform a live demonstration of how our antibody panel design tool can be used to organize the panel seen here. I have taken the liberty of adding most of these antibodies into the design tool, and I will show you how to add the subset shown here. I have my antibody design tools opened in the background, and I'm going to exit this presentation. All right, so here you can see the different tabs across the bottom. We have the summary tab, the inventoried antibodies tab, and then these 3 tabs are for you to design your custom panels. So if I go into the human fresh frozen panel, I've already taken the liberty of adding Diane's inventoried antibodies to this list, as well as her custom conjugated antibodies to this list. These last 5 columns are automatically populated, so you don't have to worry about filling them out. If you scroll to the bottom, you can see her panel currently has 5 ATO550 reporters, 3 Sci5 reporters, and 4 AF488 reporters. Now let's go ahead and add her inventoried antibodies. So we have Chi67 and pan-cytokeratin left to add. So let's go into the inventoried antibodies tab and try to find these. So the easiest way to do this is first to filter by human fresh frozen antibodies. So I'll come into this little arrow here and deselect all and only select the human fresh frozen antibodies. So here it got rid of the FFP and the mouse fresh frozen. And then I'll come into the target and I can simply deselect all and search for Chi67. All right. So Chi67 is associated with barcode BX047 and reporter RX047 with an ATO550 dye. So let's take this information back into my human fresh frozen panel and enter it. So if I go down to my inventoried antibody column and BX047, I can click in here and Chi67 is available, and it automatically filled out that it was an ATO550 reporter. So we can cross this one out. Next, let's add pan-cytokeratin. So I'm going to go back into my Inventoried Antibodies tab, and I want to clear that filter. So I can clear that and simply add pan-cytokeratin. There we go. And it brings up two options, pan-cytokeratin with an Electrophoresis reporter or a 750 reporter. And this is associated with BX019. So let's go back into the human fresh frozen panel and go to BX019. And you'll see there's only one option here. The reason for this is because it's a fresh frozen panel, it automatically assumes you're going to use 488, which is the recommendation for fresh frozen. So it automatically populates the AF488 fluorophore. All right, our panel is starting to look a little bit more even. Okay, the last two antibodies we have to add to our panel are MMR and PAX6. Now, relatively speaking, MMR is less abundant in comparison to PAX6. So for less abundant antibodies, we recommend for fresh frozen to conjugate it to reporters that are associated with the Cy5 fluorophore. And for high abundance antibodies, we suggest conjugating it to Electrophoresis 88. So let's try to find which Cy5 reporters are available and associated with which barcodes, so we can add it to this list. So let's go into our summary tab. So I want to look under my human fresh frozen column, and I want to know which positions are free. So I've identified BX006 is free, BX016 is free, BX20 is free, and are either of these positions available with a Cy5 reporter? So all I do is just go across this row, and I find that BX006 is available with a Cy5 reporter. Now I can go back into my human fresh frozen panel, find BX006, and I can see I don't already have another custom antibody entered here, so I can go ahead and decide to put my MMR in this position. And so when I go to custom conjugate my MMR, I will use BX006. And so let's enter that here. Okay, we can cross that one off the list. And finally, we have PAX, which is a high abundance marker, and we can associate that with AF488. So let's find which barcodes are available for PAX6 in an Electrophoresis 88 fluorophore. So I come back into the summary tab and I do the same exercise. So now we've already used BX006, so let's look further down. So I have this open position, next BX016. If I go across the row, I can see that yes, this is actually associated with the 488 marker. So I can choose this barcode for PAX. So I'm going to take BX016, go into my human fresh frozen panel, and find BX016 and enter my custom marker. All right, and then auto-populated the 488. And if I scroll down, you can see my marker panel is much more even. So I will have 6 cycles of markers in my multi-cycle experiment plus 2 blank cycles. So this is an 8-cycle experiment. Now, just to note, all of our barcodes are available for custom conjugation. You just want to make sure that there are no repeat barcodes in your panel. And, you know, if you want to leave these open, then you certainly can. So this is why I went to I first looked for these open positions, but if I needed to, I could always go back and choose one of these as well, so long as it's not used in my panel already. Now, thank you for joining this presentation today. I appreciate your time and attention, and I hope that this presentation will help you as you go about designing your own custom antibody panels. And with that, I am happy to field any questions from the audience.